faculty who are eager to speak already are having to be given permission to. That's because we had a lot of questions from faculty members yesterday. My name is Simon Sponberg. I'm the moderator for this session. Margie is also helping to moderate. We've got the speakers and us on the panelist side. We are going to be prioritizing student questions today because it is a student network after all. So we are going to be taking the first question from students and postdocs, and I'm quite willing to sit here in silence, awkward silence for a little bit if people aren't asking questions. So please chime in and ask questions. I really like to see that. I may not know everyone. In fact, I guarantee you I don't know everyone. So I will do my best to figure out if you're a student or a postdoc, but if you're a sneaky faculty member trying to sneak in with the first question, please defer to a student or postdoc first if I call on you. To ask a question, you can please post the question either in chat, you can type it out if that's what makes you feel comfortable, or you can use the raise hand function in Zoom. And then at the end of the session, I'll call on you. Okay, so you can just toggle that on or off as we're going through the talks. I'll be doing the moderation at the end. So again, we're going to have 7 talks this morning. This is day 2 of the iPoLS meeting. Welcome to Atlanta, sort of. We will have 12-minute talks. If the talks are going over 12 and then 3 minutes of questions, if the speaker is going into their question session and they're not like on their last slide, I may interrupt and say that they're moving into their question session. When they're done, we'll again have the questions, and that should be prioritizing students and postdocs. And then at the end of this session, which should end right around 10:50, maybe, or 11:50, a little bit later, we'll have a brief introduction to what the spatial chat feature is for doing the poster sessions, and then we'll release everyone to do the poster sessions from 12 to 1. And we really encourage you guys all to stick around for that. This is a big experiment in trying to do virtual posters. But the students and Margie have put together a huge amount of work to try and get their posters loaded up virtually. And we have a sort of cool function to allow you to interact with the posters individually. And it should be pretty lightweight. It doesn't require installing any new software. So I'll go over that a little bit more just at the end of the session. So for now, I think we're going to get started. So the first talk today is Elodie Coutinier, and from France, and she'll be giving a talk on growth and migration of individual mycelium filaments. So Elodie, go ahead and take over.
Okay, thanks. I will share my screen. Can you all see it?
You're good, go ahead. But you can go up to presenter mode. Yep, there you go. Okay.
First, I would like to thank the organizer for allowing me to talk about my PhD project. So I'm going to talk about confinement, actually. I know you've heard this word a bit too much recently, but I will talk about the confinement of Candida albicans. So maybe you already heard this name, and I will start with a brief introduction of this fungus. So it is present only in the human body, mostly in the gastrointestinal tract and on mucous membrane, and around 30% of the population have it in their organism. But hopefully this is a commensal, meaning that it doesn't do any harm, and it is present under this yeast form, as you can see here. However, it can become a very serious pathogen in immunocompromised patients, leading to severe and systemic infection. The major reason for this pathogenicity is its ability to change morphology and switch to a filamentous form called hypha. So here you can see that it has branches, and starting with a few yeast, you will get mycelium like this. Indeed, the invasion mechanism of Candida albicans involve both the yeasts that are adhering on the epithelial layer and the hyphae that are actually capable of going through the epithelial barrier by exerting huge forces. Then once they reach the bloodstream, they can easily disseminate into the whole organism under the yeast form and invade other organs. So if we look a bit more closely at the structure of this filament, they are actually composed of many compartments that you can see here that are separated by rings of septine. So the slight dark, okay. and each one of these compartments contains one nucleus. So you can see in this movie the dynamics of this nuclear division, which is quite nice. And the cell cycle in hyphae is also something we are interested in, but I won't talk about it here. Moreover, it's important to notice that this filament grow only by the tip, as you saw in the movie. One of the specificity of fungi is that they have a rigid cell wall. Due to osmotic flow, there is a turgor pressure inside the filaments, and this is actually the driving force for the growth of this filament. Combined with the fact that you have a thinner cell wall at the apex, this pressure will lead to an apical growth. And there are also a lot of biological processes inside the fungi that contribute to the delivery and fusion of vesicles at the apex in order to expand this cell wall. So today I'm going to focus on the behavior of Candida albicans under confinement. And an easy way to do that actually is to use microfluidics and microfabrication tools. So what we did was to design a chip allowing us to trap yeast in front of microchannels of various sizes in order to look at the growth of hyphae in this channel. So here is an example of channels. So this is the side view and the top view. So you have a width of 10 micrometer here and a height of 1.5 micrometer. So this height is actually a bit smaller than the diameter of the hyphae, meaning that they are squeezed in between the two layers of our PDMS chip. And so this is what we observed. So you can see here some hyphae growing in the channel. And so what we saw was quite surprising, actually, because this is not at all known behavior. Usually hyphae grow like more or less straight, but you see here like some quite nice sinusoid. And so we decided to try to investigate a bit more this behavior and to try to see what happens with different height of channel, ranging from 1 to 3 micrometer here. And we also wanted to quantify the sinusoids that we see. So in terms of amplitude and wavelengths, so we see that both of these parameters decrease when we reduce the height of the channel, meaning when they are more confined. And interestingly, it seems that the more confined the hyphae are, the more curved they are. So this means that by changing the level of confinement, we are actually able to change their trajectory of growth. To go a bit further, we wanted to see if it was possible to switch from sinusoidal growth and a normal straight growth. So the goal here is to fabricate microchannel with 2 different heights along the same channel. So the hyphae will grow alternatively in a part which is confined, then a part which is not, and again and again. So here you see some SEM images of the mold we use, where you see clearly the different steps. And so this is some movies where you indeed see hyphae growing straight here, sorry, when they are not confined, and then they start oscillating when they are confined. So here you have an example where the filament goes the other way around from the confined part, where it's doing sinusoid, and to the non-confined one. And the behavior is always the same. You have nice sinusoids in confinement and straight when not confined, again for this one. So this means that this behavior is reversible and that they really adapt to the environment. So we saw that sinusoidal growth can occur in 2D confinement in PDMS chips. But to find out exactly what triggers this behavior, we try to find other situations where it happens. Since the only mentions in the literature about this kind of oscillatory growth in hyphae was found in agar gel, we also tried this experiment. And it seems that in the bulk of the agar, so when they are in a 3D environment, the hyphae grow a bit as helices that you can see here on this confocal imaging with a staining of a component of the cell wall. Then we noticed also that at the interface between the agar gel and our solid substrate, the hyphae also grow as a sinusoid. So actually, when they reach this interface, they stick to it, and you can have this kind of 2D growth. And when changing the concentration of agar, so from 0.5% to 5%, you can see that you change the behavior. So when you increase the concentration of agar, you actually increase the stiffness of the gel. And sorry, we can make also some quantification here from these images, and it reveals that increasing the stiffness of the gel increases the proportion of filaments that are doing this sinusoidal growth, and also it increases the curvature of the filament. Exactly the same way the confinement in the microfluidic chip did. So in addition to these two cases I mentioned here, 2D confinement in chip and agar gel, sinusoidal growth can also be seen on particularly adhesive substrate in liquid medium, so without any confinement in this case. So it seems that both confinement and adhesion can trigger sinusoidal growth. So we are currently trying to investigate the contact surface and friction as a possible common parameter responsible for this behavior. But we are also looking at an underlying biological mechanism which could explain this oscillation. I already mentioned the fact that the growth at the apex occurs in part thanks to the fusion of vesicles. And actually, there is a cluster of vesicles located at the apex just before they reach the cell wall at the tip. This cluster is called the Spitzenkörper here, and its position might be responsible for giving the orientation of growth, since this is where all the vesicles are going to fuse and expand the wall. One study, for example, in another fungus has shown that disruption of these polarity complexes led to this kind of oscillation. So it could be that in our situation, the Spitzenkörper is oscillating at the tip under confinement, leading to changes in orientation and to this sinusoidal growth. So we are currently looking at the movement of this Spitzenkörper in Candida albicans thanks to fluorescent strain. So this is our very first images, but it seems that the displacement of the Spitzenkörper towards one side is correlated with growth in this direction. With this hypothesis of oscillating Spitzenkörper, we could expect that the wall inside the curvature might be thicker than outside, since it should receive more vesicles, so more cell wall material. With the staining of the chitin, a component of the cell wall, we can see that the intensity is higher inside the curve, actually. So this is something that we can quantify by taking the intensity profile of segments normal to the shape of the filament. So you can see here that the intensity outside is smaller than the one inside. And by doing that along the whole filament, we can plot a normalized variation of intensity here between the two sides as a function of the curvature. So this indeed shows that the intensity increases on the inside when the curvature increases. So I think I will stop here, and to conclude, we have found a very interesting mode of growth of Candida albicans, which can be triggered by confinement or strong adhesion. We have also shown that the trajectory of growth can be modulated by the level of confinement or by the rigidity of the agar. Oscillation of the Spitzenkörper could be the biological origin of this sinusoidal growth, and this could be correlated with the fact that the cell wall is thicker inside the curvature. But there are still a lot of questions we are currently trying to answer. We would like to determine where this oscillation comes from and how they are linked with the confinement, and also what determines its frequency of oscillation.
No.
This is Marvin Dieter. Rosemary's coming.
Okay.
Yeah.
And from a more clinical point of view, let's say, we are also wondering if this sinusoidal and helicoidal growth could contribute to virulence, because this is maybe a type of growth which is more exploratory, and this could facilitate epithelial invasion. And with that, I would like to thank my supervisor, both of my team in Institut Curie and Institut Pasteur, and also the technological platform in IPGG. And thank you for your attention.
Okay, great. Thank you, Elodie. So we'll take questions now. Please, again, students and postdocs, we are waiting for you. So Ernst Lübig, you are now able to talk. Go ahead and post your, you can unmute yourself and ask your question if you'd like. Ernst, you can go ahead and ask your question. You'll have to unmute yours.
Sorry, I pressed the button unintendedly. Sorry. And also I'm faculty, so I'm not supposed to ask.
Oh, okay. You're muted then. So we've had a couple of questions in chat. So it might be helpful for me if folks indicated if they're a student question, if you can, when you're in chat. So Zane, I think your question in the chat is going to come up first. So Zane in the chat asked, you already said figuring out where the oscillations come from, but what is your opinion? Why does the cell oscillate? What is it looking for if it's already in a good growth condition?
What is it looking for? That's
What do you think is causing the oscillation? I think?
Actually, we are wondering if, like, without any confinement, we see a normal straight growth, but it could be that you always have some kind of small oscillation of the Spitzenkörper, and it would be just like accentuated under confinement. And so this is where you would see clear sinusoidal growth. But why? That's a good question.
Okay, so Ernst, you can go ahead and ask your question now. You want to unmute yourself?
Oh, I asked Michael I unmuted Michael if he would like to ask his question from the Q&A.
Yep.
Okay, well, there's a question from the Q&A thing. How do they sense they are in a confined environment versus unconfined? Is it a steric interaction or is it something else?
Yeah, actually, something we are looking at is the contact area between the hyphae and the substrate. Because when they are squeezed, you clearly increase the contact surface between, actually, I think I have a picture of that. Like, if you look, so normally they look like that, and if you squeeze them in between two plates, you will increase the surface. And actually, you can see by doing some confocal that sometimes, if the hyphae is really thick, you really see that you increase this surface. So we are trying to look at this parameter to see if this is something that they sense. And actually, there are a lot of cell surface protein, and some of them might be mechanosensible. And we are also trying to look at some mutants of with deletion of this protein, and some of them don't exhibit this sinusoidal growth. So this could be how they sense the confinement, so the contact with the wall.
Okay, great. Thanks, Elodie. So we're going to have to move on. So again, we're going to have an open session for everybody to ask questions at the end. Yep. We'll have a special spatial chat room for them. So we'll let Elodie take more questions there. And we're going to move on to our next talk now. Our next talk is going to be Antoine Fourleau, who is from Lyon. And Antoine, you can take it over whenever you want.
Yes. I'm, oops, sorry. sharing the screen. Yo. So thank you very much for giving me the opportunity to present my work today. I would like to tell you about this method I developed to analyze signal in cellular tissues. And before that, I would like to introduce you the questioning that brought me to do such an analysis. And in my team, we are interested into cellular variability and its impact on development. So if you consider, for example, this human epidermis, it's constituted of various cells of different kinds, which are randomly distributed. But despite this variability at a cellular level, you observe that organ develops in a quite reproducible way. And my two hands, for example, superimpose quite well. They have a high degree of similarity. And the question is why this reproducibility in development despite cellular variability. And indeed, I work in a plant development lab, so I will tell you a little bit about plants. And in the team, we did a lot on this floral organ, which is called sepal, and it has the shape of a leaf. And in this organ, you can observe variability at every scale. At a subcellular scale, if you consider, for example, this stiffness map obtained by AFM microscopy of the surface of the organ, where the black lines here are the limit between cells. And what you see is that there are regions of the cells which are stiffer than others. Also at the cellular scale, if you consider this picture, for example, you see that the growth rate of cells is highly variable as well. And actually the standard deviation for this growth rate is comparable to the order of magnitude of the average growth. And also if you go to the larger scale, to the organ scale, you also observe variability. So for example, if you consider this diagram, which shows the superimposition of the organ contours for a set of individuals, you see that the distribution of shapes is distributed around an average, but the thickness of this distribution is relatively small, which indicate that the heterogeneity through development is relatively small, and we wonder why at this scale fluctuations are so small, while at the cellular scale things are so variable. And we wonder also what are the mechanisms which prevent fluctuations to invade the larger scales. And a good candidate for such a mechanism is the mechanical feedback. As I told you, the growth is not uniform. There are regions that grow faster than others. And for example, in this configuration where you have fastly growing regions on the top and below and slowly growing region on the sides, you expect residual stress to develop because the fastly growing region will tend to push the medium in the intermediate zone. And so it generates residual stress. And we know that tissues, or we think that tissues respond to such a stress. And in plants, for example, it is known that cells are able to adapt their stiffness to oppose a higher stiffness to the direction of maximal stress. We would like to know if such a mechanism is able to buffer cellular variability and finally lead to robust organ development. And to answer this question, I developed a model. It's a generic model which only accounts for the really essential ingredient in this context, which are the force balance, which drives the growth. So, for example, in plants, it is so that it is the turgor pressure which leads the growth in tensing the cell walls, and at long time scale, the cell wall relax, which allow the expansion of the tissue. Another essential ingredient is the synthesis, because as you grow your system, you will tend to dilute the components of the cellular tissue, but the synthesis is there to compensate this dilutive effect. And finally, we accounted for the mechanical feedback, so the ability of the tissues to respond to stresses and adapt their rheology. And an important prediction of this model is the existence of an optimal feedback which minimizes the heterogeneity in development. And this optimal feedback can be understood by a growth analogy in comparing things to the traffic in a motorway. And the fluctuations in the traffic will depend on how much the drivers respond to perturbations in the traffic. If the drivers have a low response to perturbation, then fluctuation will tend to be amplified, as illustrated on the bottom left of the slide, while if the drivers have a too strong response to perturbation, fluctuations will also tend to be amplified. And this is why there is an optimal feedback which minimizes the heterogeneity, well, in the motorway and as well in the tissue. Another important prediction of the model is the existence of long-range correlation in the tissue. And it means that there are signals such as, or quantities such as the growth rate, which shows fluctuations at every scale, and so show such a fractal pattern. And this is very nice prediction, but if you compare it to experimental observation, there's a sort of gap. And one possible reason of this gap is that in experiments, the information is cellularized. If I consider the growth, for example, I look at the cellular growth. And so the information is somehow projected on a cellular pavement. And the question is, if we want to show the existence of this long-range correlation, we have to understand how to analyze this kind of information, which lives on a pavement space, a paved space, yeah. And in a classical context, for example, if you want to study a time series, a standard way of doing this is to look at the Fourier transform. And as you know, the signals can be decomposed into a linear combination of Fourier harmonics, and by representing the Fourier transform, or their amplitude as a function of the wave number, you obtain a spectrum which is characteristic of the signal you want to study. This is very nice, but in our case of where we consider cellular tissues, the medium is disordered, and also in a biological tissue, the geometry is complex. Also, the information is of a different kind as the one you can analyze with a fast Fourier transform, for example. And this is why we needed to define a new Fourier transform which is adapted to cellular tissues. Unfortunately, I don't really have time to explain you what the mathematics behind this, but if you are interested in this, I think it's you will be interested by our preprint, which should be online soon. Finally, with this analysis, we were able to, with this method, we were able to generate Fourier modes. And here I represented the first Fourier harmonics. And we were able to decompose signals into a linear combination of these harmonics, and finally obtain the Fourier spectrum of signals. And here I represented you the Fourier spectrum of a field which is correlated at long range, which is why the amplitude of the lower modes are in average higher than the one of higher modes. And this Fourier spectrum has the good properties we would expect in a classical context, even if, well, I don't really have time to explain you what I mean by this. And this method is quite general, so it can indeed be applied to all sorts of disordered material geometries, and also it can be used to do statistical estimation, as I want to do in the tissues, but we could imagine to use it to do pattern recognition or other things. Finally, I applied this method to experimental data. And so I looked at the growth of the sepal, this floral organ, and I decomposed the growth field into Fourier harmonics. And by doing this, I was able to represent the Fourier spectrum of the organ. And here I represented two Fourier spectra, one for a normal individual, wild type. And you see that the Fourier spectrum is more or less flat, which indicates that the signal is correlated at long range, at short range, sorry. While if you consider the same spectrum, but for a mutant, FTSH4, you will see that the spectrum is quite different, and this time corresponds to a statistical signal correlated at long range with lower modes which have higher amplitudes. And the reason why I show you this peculiar mutant is because it is known to develop in a more heterogeneous way. And so all this is preliminary results, but we really believe that by applying our analysis to all sorts of tissues, we should be able to really investigate the mechanism at play to regulate fluctuations in tissues. And also we hope to infer the rheological properties of the tissues, because as you may guess, fluctuations are related to relaxation in the tissue. And to conclude, I would like just to say that I think I defined a really robust and well-defined mathematical method to analyze signals in cellular tissues, and that by applying this to different tissues and different signal, we could learn a lot on the physics of these tissues. And finally, I would like to thank all of you, and also Areski, my supervisor, and the institution who provided me financial and material support. Thank you.
All right, great. Thank you, Antoine. So again, we'll open up the question floor now. Now I am monitoring the question and answers and the chat, so I should see all three possibilities. So if students have questions that they'd like to follow up on, please post them up. And the questions from the previous talk have been moved to the answered section, so it's clear there. Okay, so Thomas Day asks, very cool ideas and talk. While I am sure that there is mechanical feedback to the plant development, couldn't organ level variability stem from cell level variability, just from propagation of error? How have you ruled out this kind of variation?
Sorry, I didn't get the...
So the question is posted in the Q&A. You can look at it, but I'll repeat it as well. So Thomas Day asks, while I am sure that there is mechanical feedback to the plant development, couldn't organ level variability stem from cell level variability just because of propagation of error? How can you rule that out?
Yeah, so in a, let's say in a classical system, you would expect the fluctuations of the big system to scale like 1 over the square root of the fluctuation, the small fluctuation of the size of the system. Let's say it's the standard central limit theorem. But here, because of growth, you have that growth tend to expand the fluctuation and tend to so it's, at least it's what we believe. We think that because of growth, the fluctuations tend to be much more, much bigger at large scale, and that's why we predicted these long range correlations. And so yes, the fluctuations at small scale, at big scale, originate for the ones at small scale, but depending on how the tissue respond to the source of fluctuation, the propagation is more or less strong, I would say. I'm not sure if I'm being very clear, but yeah.
And then a follow-up question from Isabella is, I was wondering how well you need to know the disorder of the material or the geometry in order to be able to use the redefined Fourier transform reliably.
So that's the thing. So that's why we define this Fourier transform, is because we want to study the fluctuations of growth, for example, but there are other things which fluctuate, such as the cell size, and also the arrangement of cells varies, etc. So we needed to really define a method which gets rid of all these or account for all these other source of fluctuations, so that it doesn't perturb the analysis. And I think the analysis we get is quite good for that, in the sense that we really are able to get rid of all this bias due to for example, variable cell size or this kind of variable cell pavement geometry, etc. Yes.
Okay, great. So unfortunately, we're going to have to stop there with the questions. There are at least one more question posted in the Q&A, and speakers, you can go and look at the Q&A. All the unanswered questions will actually be moved to the answered session at the end of your talks, because we'll want to leave it clear for the next talk. But you can take a look at those and answer them. You can also discuss them afterwards. So we'll move on now to the next talk, after thanking Anton again. So Kanish Jain from Emory, from Gordon Berman's lab, will be giving the next talk, and Kanish, you can take over. And Kanish, you're still muted.
Okay, sorry about that. Hi, everyone. My name is Kanish Jain, and I'm a graduate student here at the Berman Lab University. And today I want to talk about methods to represent rat behavioral dynamics. So at Berman Lab, think about how to—
I'm sorry, Kanish, I have to interrupt you. The screen share setting you're on is optimizing. Ah, there we go. I think it jumped in. So I think it's clear for most people. People are having issues seeing the resolution. Let me know in chat. Sorry for the image.
Okay, it should be better now. Okay, so here at Berman Lab, we think about behavior automatically represented a lot, and behavior multi-scale process is it's a challenging problem to do that. So it can be driven by internal mechanisms, hunger and anger. It could be repetitive postural movements. It could also be interactions with friends and foes, in this case, in the environment, and it could be learned behaviors learned over generations. So how can we quantify such behaviors? We know that quantifying free behaviors would be central to understand the underlying neurobiology that is driving such behaviors. So what numbers can we put on these? Maybe the weight of the pizza or weight of a rat, or the velocity of the dung in this case, or maybe a scorecard in this. We can do better. So to address this challenge, we work with this data set taken by Elena Menicini and Tommaso Mutu in Dr. Aman Salim's lab at UCL London. This is a 3-hour data set, and it's part of a bigger data set, which includes simultaneous neural recordings as well. And we are trying to understand perception and free behavior. But for the purposes of this talk, we'll talk about how we are trying to quantify free behavior first, and then we will look at neural correlates later for studies. So to begin with, we use a homebrewed version of a markerless tracking algorithm called DeepLabCut. We track around 14 points on the rat using DeepLabCut, and ego center these track time series so that the rat is always pointing right. The tracking is almost good, other than at places where there are occlusions, such as when the nose goes under the rat's body, but otherwise the tracking is good. Once we have structural tracking, what can we do with it? Well, since we are at Berman Lab here, and we have this pipeline developed by Berman et al. In 2014, use the time series to create to use in these pipelines, which I don't have the time to go into the specifics, but in and then embedding these spectrograms onto dimensional stack maps. And when we do that with these postural time series, we get this map. If you're not familiar with the behavioral density map, you can imagine each time point in the postural time series being a point embedded in this space. And then the colors here represent the probability of those points in the space. So is it even meaningful? Well, we can look at the velocity map, which shows the velocity of the on the right in the arena. And we see that high velocity regions here are clustered together on the top right here, which is interesting because we put a percentile time series and it's able to cluster all the active behaviors together. So this is a little bit promising. We can also look at each of the peaks in this map and try to look at the frames in these peaks and annotate behaviors in each of these peaks. And that's what I do here and show on the right. We see different colors on the right, which are different active behaviors, and then a lighter shade of blue and gray on the left, which are mostly idle and sleep behavior. Here are some examples of these behaviors. On the light blue here, we have sleep, eating, where the rat is chewing on, locomotion in the red here, and rearing in the orange here. I should also mention the arena has a lot of toys, a lot of food, and a lot of water, since it's a 3-hour-long recording. So we see a lot of different behaviors. So I also mentioned there are these gray here, unknown or gray areas, if you may, and which include a lot of non-stereotyped behaviors, which I was not able to cluster. So there's an issue. There's one we have. There are also some gray areas on the outline here, which are mostly tracking errors coming from deep lab cut, which are not being represented in the map itself. So there are some drawbacks here, and if we compare this map to the fly map from the 2014 paper, we see that there are not a lot of features shown in this map. There are not a lot of peaks in this map, and that may be because rat behavior is not as flies as we would see. It could also be because of correlated postures in rat. So there's one way to address this challenge is if we are making a generative representation instead of a movement-based representation, which we make here. And to make a generative representation, we need a dynamical model, and it might help us uncorrelate the and all denoise the time series at the same time. And one cheap, effective, and quick way to get a dynamical model is using neural networks. And we can train a recurrent neural network to predict postural time points at the next time point given the current time point. And since there is recurrence, we effectively get a dynamical model, which is over-parameterized, of course, but it predicts the next time point posture based on the postural time series from before. And if you are an aficionado, I've used LSTM units in these RNNs. So once we have this model trained, we can use the internal states, which are the hidden states inside the RNN, to create a representation which would give us a more generative picture of the rat behavior. And we can use these internal activities and put it in the same pipeline, which I showed earlier. And this is the map we get from this pipeline. The map on the left, you see, has more features right off the bat compared to the map we got before, which is on the bottom right here. So we like to see more stereotypic behaviors and more features in this map. And let's try to quantify that. We can look at the velocity of the rat in the arena here and map that following the same line of investigation we did before. We can also look at the body curvature, which is 0 when the rat is straight versus some value when the rat is curved when sleeping. We see there's more curvature here, so this should be sleep, and these here on the top are active regions. We can again look at the peaks and annotate behaviors. And here are some examples of the behavior, starting from the green region, eating here, the red region, locomotion here. There are some yellow regions here, which are head turns. And then there are some regions here on the blue, which are just head movements. So it seems like we are capturing stereotypic behavior. You should also notice there are less gray regions here, which is because we see more stereotypic peaks here. So, okay, next slide. Okay, so we get this map using RNNs and it works well for us. But can we harness deep learning tools, other properties of deep learning tools for our benefit, for uncovering more things about from such models? Well, we can look at other deep learning tools such as CNNs, convolutional neural networks, in which features, as we go into more and more depth, become more and more complex. They start out with just lines, looking out for just lines, then move on to circles, then more and more complex features as the depth increases. So can we do something similar with RNNs? So yes, we did. And we tried a number of different architectures, but for the purpose of this talk, I will show a 3-layer simple example here. In train a 3-layer RNN, predicts the next postural time point, given the previous postural time point, same scheme as and now we can make a separate map for each of these hidden activities in each of these 3 layers. And this is what we get. So you can see the first layer representation is very similar to the 1-layer representation we get. But as we move on to the 2nd and 3rd layer, the representations are more clumped together. But are they even meaningful? We can look at the velocity of the rat, same as we did before, and we can see that, yes, there are some active regions here in this map, here in this map, here in this map. So there is some representation still, and even though they are clumped. And if we try to, again, annotate the peaks in these maps, we see that, yes, they do make sense. There are some stereotypic behaviors in all 3 peaks, even though they are so vastly different visually. So here are some examples from layer 1. We see head turns here. We see locomotion, some head movements here. And these are the layer 3 layer frames from layer 3. We also have locomotion, grooming, eating, and rearing. So even though we have such functions in all of the 3 layers, we get similar stereotypic behaviors in the peaks. So what is different here? What is the secret sauce that's making them so different? Well, maybe we can look at the distribution of points. If we look at one peak in layer 1 and see how they are distributed in layer 2 and 3, we see that layer 2 and 3 are more clumped together. Here's another example. But if you look at a peak in layer 3, we see that it's more distributed in layer 1. Here's another example. And we can quantify this disproportionate distribution points peaks using this conditional entropy-like metric. And I have to skip that slide to save some time, but the numbers do agree with what I'm trying to show here. So, okay, moving on from this data set, can we apply RNNs to other data sets? So I showed this map from the 2014 paper before, which was made using the image of flies. And thanks to this paper from 2019, where they use a tracking algorithm a developed tracking algorithm to track joints on these flies. And if we make a map using a movement-based representative map based on this tracking, we get this map if we use angles versus joint positions. And if we compare it with the map from before, you made using just images, we see that there are very similar topological features in this map. We can see locomotion here in the same area. And if you stare at these long enough, you will see that you will be able to pick out same peaks across maps. While does it do any better? Not really. It kind of gives us the same representation. So it seems if we have a fairly uncorrelated data sets, RNNs don't give us as much of an edge as if we have a correlated data set. Another example of a correlated data set is this motion capture data taken by Dr. Jesse Marshall in Dr. Benzawiecki's lab at Harvard, where they track 3-dimensional joints on the rat using motion capture. And if we train a 3-layer RNN, this is the presentation we get. So it seems like this method works very well with these uncorrelated correlated data sets. With that, I would like to thank you for your attention. I would like to thank my advisor and collaborators. I would also like to mention that if these methods may be useful to you, please look out. We will be releasing a Python open source implementation soon. You can follow my advisor on Twitter, and you can get the information there. Thank you.
All right, great. Thank you very much. So we'll again take questions, again, students and postdocs primarily, if you have questions. We have one coming up from Nathaniel here. We'll assume you're a postdoc or a student. Were the maps annotated with the corresponding behaviors afterwards by a human, or were the behaviors characterized before the model was trained?
The maps were annotated afterwards. These were, maybe I forgot to mention, but these were 3-hour-long recordings. So there's 50 hours of total behavior, and it's very difficult to annotate it manually. So they were annotated afterwards, after the peaks were determined and the frames in the peaks were determined.
Other questions? So you have another question now. Why is most of the activity of the rat confined to the one side?
Most of the activity could be, so if you're asking why idle frames is so big, it could just be because there are a lot more frames for idle, and there are a lot more postures for idle. So if you look at a whole 3-hour recording of the rat in the arena, you would see that it's active for mostly half an hour out of the 3 hours. So it's sleeping for most of the time in the arena.
Can you go into more detail about what makes the rat data different from the fly data, and why the RNN maps provide more information for the rats?
Yes, absolutely. So if you're, you can see the joints on the flies are very uncorrelated. Whereas if you go to if you recall the tracking on the rat, you see that the joints that we are tracking on the rat are more correlated. So that could be one reason why the maps for the rat are, the RNN maps for rats are giving more information, because RNNs as a dynamical model are able to decorrelate these movements much, much better compared to just directly using images of rats.
And then how do you know how many levels of the neural network to use in order to get meaningful behavior? Why not 4 or 5 layers?
Yes, we can use as much as we want, but qualitatively, we have done that. We have gone up to 6 or 7, but qualitatively, the maps do not change as much after 3 or 4 layers. So yeah, there's no upper limit. You can train as big as you want. The only challenge posed there would be maybe some gradient challenges while training, but other than that, nothing stops it.
Okay, great. So let's thank Kanishk again, and we'll move on to our next talk, which is Ahmed Roman from Ilya Niminen's lab at Emory. And Ahmed, if you want to share your screen and start, you can go ahead. Oh, looks like we've accidentally dropped Ahmed here. RG, do you have Ahmed in the? So Ahmed is not.
Oh, there he is. Here he comes.
Okay.
Yeah, I got kicked out. Okay, so
go ahead, Ahmed.
Okay. So hello, everyone. Thanks for giving me the opportunity to discuss my research. So today I would like to talk about modeling learning and animal behavior in C. elegans. So once I get rid of this, okay, so there we go. And so this work has been done in collaboration with Konstantin Plansky, Ilya Niminman, and Will Ryu. Ilya is my advisor, and Konstantin and Will are the ones who actually did the experiment in Toronto. And just to give a little bit of motivation, so there was a paper that was published in 1973 by Hitchcock and Russell that showed that when worms are reared at a particular temperature with food, then the worms will thermotax to the temperature, to that temperature. And I give a few examples in the top there. So you see worms that were grown at 15 degrees on the left and 25 degrees on the right. And you can see that they report on a temperature gradient from 18 to 22 degrees, and you let them move freely. And then the worms that are grown at 15 degrees, you prefer the colder side, and the worms that were grown at 25 degrees, they prefer the hotter side. They further showed that if you starve these worms at a particular temperature for a few hours, then they will run away from that temperature. And that exposure to a new temperature for a few hours can actually shift the preferred temperature of the worm. And finally, in 2006, David Byron actually showed that there is a memory molecule, which is an epsilon kinase, allows the worm to integrate prior thermal temperature. It actually allows the worm to compute a weighted average of the temperatures that it has experienced before. And so that temperature, that weighted average of all the temperatures it experienced, is the preferred temperature, or the temperature that it prefers now. And so our goal is to actually model this behavior and hopefully make a few predictions in the process. We want to model all of these various components of learning and hopefully make a few predictions in the process. So first, there was this really novel experimental assay that was developed by Will Ryu and Konstantin Polansky, which I will go through right now. So first, you get nematodes, which are cultured at 15 degrees or 25 degrees. These worms are taken 6 at a time, sequestered in 4 millimeter droplets. And these droplets are defined by printed Teflon slides. So you can see on the left here, there's the printed Teflon slide with all of these circles where the droplets would go. And each one of these droplets is about 4 millimeters across. Then this assay is sealed and is imaged at 6 hertz. Each one of these droplets takes a single worm. And then a temperature gradient, a linear temperature gradient on each worm, which is about half a degree Celsius per centimeter, occurs from the top of the droplet to the bottom of the droplet. So the top of the droplet is 20.4 degrees, and the bottom of the droplet is 19.6 degrees Celsius. And provisionally, we just define these to be +1 and -1. And so the middle temperature, which is 20 degrees, is just 0. And then the center of mass position is actually tracked by, of each worm is tracked with the CC cameras. And then the thermotactic index is then defined as the average position, center of mass position, over 60 worms. And so in the bottom here in panels D and E, you can see 2 examples where the worm is athermotactic, so it doesn't care which temperature it goes to. And another example where the worm is actually cryophilic, so it prefers to be near the bottom of the droplet where it is colder. So now, once you actually do this averaging over the 60 worms at least, then you can get these really nice trajectories for the thermotactic index as a function of time, and these are 4 hours long. So you see here on the left and on the right, the worms which were well fed at 15 degrees or 25 degrees respectively, they show initially this cryophilic or thermophilic tendency. So the moment you turn on the gradient, so in the beginning, there's a 15 minutes of no gradient, and then you turn on the gradient, and then immediately the worms that were raised at 15 degrees go towards the colder side, and the worms that were grown at 25 degrees go towards the hotter side, which is exactly what we expect. But then the droplet contains no food. And so for a few hours, there's no food in the droplet, and the worm decides to change its preference and go towards the hotter side or the cold side respectively. And so this non-monotonic behavior indicates that there are multiple dynamic processes with multiple distinguishable time scales that the worm is using to actually produce this behavior. So now we need at least two dynamic processes. What we mean by dynamic processes is two autonomous differential equations. If you're going to model this, you need at least two differential equations to actually model this behavior. But upon further inspection, you can take these curves for the thermotactic index, which we could say they are just a function x of t, x as a function of t. And instead of plotting the thermotactic behavior as a function of time, we can plot it as a function of itself. What I mean by that is you take that trajectory x of t and plot x of t minus 1 versus x of t. So it's the thermotactic behavior is just retarded by one time step. And so you get these delayed embedding plots. And the point here is that if these curves intersect, then we know that actually two dynamic processes are not enough. Two differential equations are not enough. You actually need at least three. And you can see here on the left, the well-fed lines do not show any intersections, but on the right, the starved lines actually show a lot of intersections. And so consequently, we know that we actually need at least three differential equations. And this is just to begin. We want to know how many differential equations do we need in order to model this process. So now we know at least we need three. But if we look at the 25 degrees data, the well-fed line here is in pink and the starved 30-minute line is in red. And you see here that these two trajectories live in the linear regime. And they oscillate at different frequencies. So the well-fed line needs 3 hours to make a complete oscillation, while the 30-minute starved line, of course, is starved 30 minutes at 25 degrees before it is put in the droplet. So that oscillates, requires a much longer time in order to complete an oscillation. But we know from eigenvalue analysis, we need that in order to get an oscillation frequency, you need a complex conjugate pair of eigenvalues. That gives you a single oscillation frequency. So if you want multiple, if you want two oscillation frequencies, then you need at least two complex conjugate pairs, which means we need 4 differential equations. So we thought we needed 2, and then we thought we needed 3, and now we actually need 4 differential equations in order to model this behavior. So here is the model. So the first equation describes the habituation of the worm. So the worm is conditioned to, is grown at a particular temperature, and then it grows to like that temperature or habituates to that temperature. So that first equation integrates over all the temperatures that the worm has experienced. And so it gives you a weighted average of the experienced temperatures in the droplet. The minus h term gives you the extension. So if you're not experiencing that temperature, then you should not be habituating to it. Your habituation should decay to 0 for that temperature. And then there is a generalization with a term which is H15, which basically says that your experience at the training temperature or the culturing temperature, so if you're grown at 15 degrees, your habituation there should influence your habituation at the new temperature. And we have another equation here, which is for the starvation avoidance. And it integrates over all the temperatures at which there was no food. So that basically it's integrating over all the temperatures at which the worm is starved. So that's the first term. The second term is again extension, and the third term is generalization from the starvation avoidance memory that you had at 15 degrees. And then of course the 15 degrees habituation and avoidances are just decaying exponentially because the worm is no longer experiencing 15 degrees in the droplet. It's at 20 degrees. And the preference, the temperature preference of which side the worm is going to go to is just a sigmoidal function of the difference of these two memories of habituation and avoidance at 20 degrees. Okay, so now once we have this model, we would like to train it. So here we have 6 lines, a well-fed or starved for 1 hour at 15 degrees, starved for 2 hours, and etc. The dashed lines are the experiment, and the solid lines are the theory. And so this is the thermotactic index as a function of time. And you see here that the well-fed lines behave as expected. You turn on the gradient, they were raised at 15 degrees, so they immediately go to the colder side, which is the bottom curve. They go towards the bottom. But you can see here that if you starve these worms before you put them in the droplet, in the gradient, that this tendency to go to this preference actually starts to change. And by 2 hours, the worms that were raised at 15 degrees, because they were starved for 2 hours at 15 degrees, they immediately go towards the hotter side, not the colder side. And so from this data, we can actually learn to fit all of our parameters. We had a bunch of parameters here. We had the habituation time scale, the avoidance time scale, the habituation and avoidance amplitudes, and also time scales for habituation and avoidance at 15 degrees. And so once we learned all of these model parameters, we would like to actually validate this model. We would like to test this model. And so we had a mutant that was not used in the training of the model, which is the INS1 mutant. It's an insulin mutant that basically kills off the avoidance, the starvation avoidance behavior. It keeps the habituation, but it completely destroys the avoidance behavior of the worm, the avoidance response. And so if the model is correct, then just by letting the avoidance amplitude be equal to 0. So again, that basically this second equation, the acquisition term would be 0. If we just kill off that term, then we should be able to reproduce this curve without any fitting at all. So that's just a prediction. And indeed, you can see that theory actually predicts the behavior of the insulin mutant quite well. Another mutant that we used here is the age 1 mutant, and that changes the time scale and amplitudes of avoidance response. And the consequence is that it introduces this multiple oscillation lobes, which again confirms that you need multiple dynamic processes in order to actually capture this behavior. And so if we keep all the learned parameters fixed and only allow the time scale and amplitude of avoidances to actually vary to fit this curve, we should be able to actually fit this curve. And indeed we can. And so again, this confirms that the age 1 mutant, it only affects these two parameters. Again, in summary, so we had this novel experimental setup that tracks the temperature preference of the worm with a precision of 100th of a degree Celsius at very high temporal resolution of 6 Hertz and for 4 hours at a time. And what we observed is that worms behave as expected. If they're grown at 15 degrees or 25 degrees, they go to, they exhibit this cryophilic or thermophilic tendencies respectively, and starvation during the conditioning or training at 15 or 25 degrees, or in the droplet, reverses these tendencies. And this preference reversal indicated that we need multiple dynamic processes with multiple distinguishable time scales. So we built a continuous reinforcement learning model that accounts for these multiple time scales and allowed us to make predictions about the behavior of the mutants and actually detangle these mutants and pin their function by using this model. With that, I'd like to thank Ilya, my advisor, and Will Ryu and Konstantin for the experiment, and the NIH for funding me, and NSF for funding the experiment and the project. Thank you very much.
Great. So we're run a little bit into the question session, but if there's a quick question or two from students and postdocs especially, please post those up now. And you guys can also post questions at any time during the talk, and we'll just save them for the end. It's actually helpful to have them earlier. So if something comes to mind, please don't wait until the end. You can write them up at any time. Any questions here from Ed? We'll wait just another few moments. All right, since we're getting close to the next time, I think we're going to go on to the next talk, but Ahmed will be available afterwards as well for doing questions. I know there's some faculty members holding their questions to try and give space for the students. So there'll be a chance for more questions and answers afterwards. So Bashi, if you can go ahead and queue up your talk, and Ahmed, if you can stop sharing your screen, thank you. So our next talk will be Bashi Chong from here at Georgia Tech in Dan Goldman's lab. And Bashi, when you're ready, go ahead and take over. And remember, folks, that we are trying to prioritize questions. That's an important part of this. So please try and engage with questions at the end of the talks. Thank you. Go ahead, Bashi.
Sure. Hello, everyone. Hello, everyone. I'm Bashi Chong, and today my talk is geometry mechanics, rationalized undulatory template modulation across scales. So the lateral body undulation is a very important template for limbless locomotors. The lateral body undulation is observed in the micro scale locomotors such as sandfish, snakes, sidewinders, as well as the micro scales such as C. elegans. And as we know, that lateral body undulation is well-known template that drives forward motion. The turning motion for the limbless locomotor is not as intuitive as the forward motion. So here, that's two examples of the turning motion observed in limbless locomotor. The first one is the omega turn observed in C. elegans. This video is taken on agar. Another one is the sidewinder snake on sand. It's the reversal turning. So yeah, see here. These are very interesting turning behaviors. Although there are too many differences between the size, among the size, the substrates, the interactions, there are still similarities between these turning behaviors. That is, the friction forces dominate the kinematics of the turning. So that means that the locomotion is almost only depends on of shape changes. So that means the motion is kinematic. So we call it geometric motion. For the geometric motion, there is a framework called geometric mechanics that we used to find the connections between the changes in the shape space to the changes in the position space. So here's an example: a swinging swimmer. It goes through all the changes in this shape space and leads to a display in the word frame. And this vector field is established connected by the force and torque balance. The ground reaction forces is by resistance force theory. Law, the line
Bashi, maybe turn off your video feed.
It's off already. Bashi, you had, oh, looks like we may have lost Bashi here. So I think that there was a bit of a gap here. So while Bashi's getting back on, what we're going to do is we're going to reorder the talks here a little bit, I think. I think we'll let Bashi reset his connection. And if he comes back on, we'll put his talk at the end. So Albert, if you're ready, I think we're going to go ahead and move on to your talk, and then we'll come back and let Bashi pick up where he left off.
Yeah, sure. Sure,
So,
I'd be happy to do that.
Albert, if you can, oh, Bashi may be back now. Let me just see if he's, Bashi, are you there? Can you get started again quickly here? All right.
Sorry about that.
Let's give Bashi one more shot here. Albert, thanks for that. And
yeah, no problem.
Bashi, we missed a bit of the last of slide 6. You sort of cut out right at the beginning of slide 6. So if you can pick up there again and try and make sure you're done in time, we'll keep going.
Okay. Geometric mechanics is the framework we use to establish the relationship between the changes in the shape space to the changes in the position space. So that means here's an example of a thrilling swimmer. It goes through a sequence of shape changes, and and it will lead to the net displacement in the work frame. So by establishing this relationship, we need a ground reaction forces. In this project, we stick to the isotropic Coulomb friction. So by Stokes' law, Stokes theorem, the line integral along the path in the shape space in the vector field equals to the area integral of this curl. So in this way, we can just design gates by finding a zero contour in the height function. So that is our tools to study the geometric motion. So with these tools in hand, we can study the turning behaviors in C. elegans. So here's a video of C. elegans turning, and we track it by deep lab cut, and it works well. And we obtain the body curvature profile of C. elegans performing the omega term. The y-axis is the body length, x-axis is the time. So you see that there's a little bit reversal here. We do the PCA analysis on the body curvature profiles for the 7 individuals, 11 rounds. And you will see that this is 4 PCs that captures the most of its variance. If we take a further look at this PC, the 3rd and 4th PC corresponds to the long wave traveling wave that drives the forward motion. So that is the PC you would observe in the forward motion of the C. elegans. And the 1st and 2nd PC corresponds to a short wave traveling wave, only appears during the omega term. So the interactions between these between these 2 traveling waves is the reason that appears to be the reason that the C. elegans can perform omega term. And with these 4 principal components, we apply the geometry mechanics tools, the amplitude modulation in the long traveling waves, the amplitude modulation of the short wave traveling wave, how the long wave and short wave coordinate with each other. So here is the a big picture. So as a result, we optimized for a gate and implement it on a robot. So here's the robot implement the optimization of prediction from the geometry mechanics. So it turns out that this gate is working well. And so here's a little bit the insights into how it turns. So there's first initialization of the turn. So the long wave that drives the forward locomotion, the amplitude goes back to 0. And while there's a superposition of the short wave traveling wave. And there's very interesting reversal here. The amplitude of the long wave traveling wave, the amplitude goes to negative. There's a reversal going on. And after the turning, there's a completion of the turn. The short wave amplitude goes back to 0. And the long wave amplitude goes back to the normal values that drives the forward motion. So if we compare the snapshots of the warm term and the robots from geometry mechanics prediction, you will see that the process of omega term is pretty much similar between the worm and the robots. And not only that, we can also capture the body orientation, time evolution of the body orientation, and it compares well with the theory. So in this way, to summarize our ideas for the understanding the kinematics of omega turn in C. elegans can allow us to discover anchors beyond the macro scale locomotors, such as snakes. And talking about the snakes, sidewinders, the sidewinders also have 2 traveling waves coexisted, but they're in different planes. One in the lateral plane, the horizontal plane, and the other one in the vertical plane. So properly coordinated vertical wave and horizontal wave can enable the lateral translation. The modulation of the horizontal and vertical wave is required to facilitate the turning behaviors in the sidewinders. Here's an example of the sharp reversal turning. And we use the similar ideas that we discussed elegans. The x-axis is the being the phase of the horizontal wave, y-axis being the vertical wave. So we can use the geometric mechanics height function to predict this motion to facilitate the turning behavior. So here's the result of a robot performing the turning by modulation of the lateral wave and vertical wave. So to summarize, the geometry mechanics, the geometry we can use this as a tool to understand the principles behind animal locomotion. And geometry mechanics is also guides the gate design process for the robot locomotion. And finally, the robot can help test the hypothesis animal locomotion. On the other hand, animals can provide templates for robot locomotion. So in this way, we have coming along with geometry, biology, and robot coming up nicely. And in this way, we can have better understanding for each case of the system. That's my presentation for today. Thank you.
Excellent. Good recovery, Bashi. Bear with us on the technical problems. Thank you all. So now if anyone has questions for Bashi, please go ahead and post those up. And again, if you'd like to ask your question, I am monitoring the attendees for raised hands, although it's easiest if it comes up in the Q&A. We've still got a few minutes for questions, so. So Ahmed asks, are the dynamics time reversible?
Yeah, so if you reverse the time, it will turn in action. It will go from clockwise to counterclockwise or otherwise.
And now, since we had a student question, we can have a faculty question. Where do the assumptions of this analysis break down? Can I also apply these to slithering humans?
Yes, so there has been studies of slithering snakes of applying the geometry mechanics. I believe that is one of the Jennifer Reeser, our postdoc in our group, that is primarily her project.
And then Silke asks, what are the rules that determine the robot worm movement? What is optimized?
That is a subject that is according to our choice. So in this presentation, the objective is to optimize for the turning angles per cycle. And we can also choose it to be the energy per, the turning cycle per energy, per energy, or displacement. We can choose that.
I noticed that the robots have a tail attached to one end. Any way to get rid of that, or is that functional? What is that?
That's not functional, but that the robot was used to build to search and rescue. That tether was used to pull the robot out when if the robot was stuck in the barriers. So.
To rescue the search and rescue robot. Is it possible that the decomposition into long wave and short wave dynamics is an artifact of using linear dimensionality reduction approaches like PCA?
Actually, so this is a very interesting question. So the long wave and short wave that appears that might not be the artifacts. That is because if there's only one wave, there's no turning behaviors that it can perform. So it has to be two waves. And especially if we compare with the dynamics of these trajectories in this four-dimensional shape space, you will see that the worm, there's an agreement between the worm turning and the geometry mechanics prediction. So it might not be an artifact. However, it should be some of the principle that guides the modulation.
All right, great. So I think that we're going to end there. Thank you, Bashi. That was great. Thanks all for chiming in with questions. Usually if we just sit here for a moment or two, they start flowing in. So that was good. So our next, so we'll go on and go ahead to our next speaker. So Bashi, if you want to drop your screen down, and we'll have Albert Lin from Ravi Samuel's lab at Harvard. Albert, if you want to take over.
Yeah, can everyone hear me and see my screen?
Yes, now it's gone. Yep, all good. Go for it.
Great. All right. Good morning, everyone. So today I will be discussing one of our projects in the Samuel lab, conducting whole brain functional imaging of the nematode C. elegans, which we've heard a little bit about already today, with deterministic landmarking. So C. elegans has long been an attractive system for neuroscientists, mostly because of how compact its nervous system is. So the worm only has 300 neurons, and the synaptic connectivity of C. elegans has been entirely mapped. So each of these 300 neurons has a name and a lineage, and C. elegans development is relatively stereotyped. These are all things that make it a great model system to study compact circuits. The neurons in C. elegans are loosely organized into 3 layers: sensory neurons, interneurons, and motor neurons, and we can gain optical access to all of these layers. So, here we go. So, identifying these neurons when imaging in vivo, however, remains a challenge. So, neuroscientists have long made progress by expressing indicators of neuronal activity, such as GCAM, in single neurons or small subsets of neurons. However, with sparse labeling like this, we cannot observe what is going on in the rest of the brain of a single animal, resulting in an inherently restricted view. More recently, pan-neuronal imaging with cellular resolution has become possible, giving us incredible insights into brain-wide dynamics. However, when relying only on cell shape and position, only a fraction of the recorded neurons can be confidently identified. Without IDs, we're limited in what we can do with pan-neuronal data. We cannot reliably compare data across animals, nor can we make direct comparisons between functional activity and the C. elegans connectome. A tool which landmarks all neurons in the animal would overcome these limitations. It would allow us to span these 2 scales of inquiry, interrogating complete circuits simultaneously in a single animal. And developing such a tool is exactly what this project set out to do. So NeuroPal is a deterministic multicolored labeled line developed by our collaborator at Gemini in Oliver Hobert's group. So what does deterministic and multicolored mean? Well, every neuron in the animal receives a color barcode, which is consistent across animals. And this color allows it to be distinguishable from its neighbors. We can therefore build a color atlas, which allows for all 300 neurons in C. elegans to be uniquely identified. 4 distinguishable fluorophores were used to build this line: BFP, PSI-OFP, RFP, and M-Neptune. These fluorophore choices leave the green emission channel clear, and this allows NeuroPal to be crossed with reporters like a pan-neuronal GCAM, which can read out neuronal activity. So our role in this project was to optimize the landmark string for functional imaging and to develop methods and software for associating neural identities with activity traces. In these experiments, we've used a microfluidics device to present immobilized animals with 3 chemical stimuli that have been previously well studied in C. elegans: sodium chloride, butanone, and pentanedione. When imaging the animal inside the microfluidics device, we would first acquire a multicolor volume, which we use for landmarking, and that's what you see here. Then we recorded the GCAM channel alone at a speed of 4 for the duration of the experiment. So you can see here the overlay of the GCAM channel and neurons getting activated. Stimuli were presented in 10-second pulses in a randomized order with 50-second buffer blanks in between. The process of assigning cell IDs, by the way, to all these colored neurons was initially done manually, but it's now being automated by Liam Paninsky's group at Columbia. We recorded from a total of 21 animals, capturing over 100 neurons in the head of the worm. So when analyzing these data, we first began with a sanity check, comparing some responses to the ones we see in the literature. And we focused first on the well-studied sensory neurons ASE and AWC. And we find that we recapitulate the known responses of these two neurons to these odors. We also observe, however, these neurons responding to the other stimuli. We see ASE get activated by butanone and pentane-dione, and AWC is inhibited by sodium chloride. We also find that a number of sensory neurons respond reliably to these stimulus presentations, a subset of which is shown here. And so while a handful of these neuron responses have been previously reported, most of them are novel findings. Similarly, at the interneuron layer, we also capture many stimulus-evoked responses, which display diverse response dynamics. The traces that I've highlighted are just a small fraction of those we collected as part of this data set. Here we can see all of the sensory neurons in the head of the animal, and each of these columns here records their peak average activity in a period before salt delivery, during salt delivery, before butanone delivery, during butanone delivery, and the same for pentane-dione. We captured many intermotor and pharyngeal neurons as well, a total of 107 neurons with sufficient N across 21 animals. Of these, we see about 30 neurons responding significantly to each of the chemical stimuli, representing a broad subset of the total. These subsets of activated neurons are stimulus-specific and partially overlapping, and this suggests that stimulus identities may be encoded combinatorially, not just at the sensory level, but also the interneuron level. Note that the pan-neuronal survey also captures neuronal activity which is independent of stimulus delivery, so things like cyclic motor circuit activity. Analyzing the data at a network level, we computed the pairwise correlations for all pairs of neurons in our data set. And I want to emphasize here that only with neuronal IDs is it really possible to build a representation of reliable network response to stimulus, because these IDs enable us to average across animals and therefore separate stimulus-related responses from other. So each box on this heat map represents the correlation between the activity of two specific neurons, and that correlation is being averaged across animals. We see that during a stimulus presentation, so in this case, sodium chloride, we see a large number of neurons get activated in concert. And if we switch from sodium chloride to another stimulus, so this one's butanone, plotted with identical ordering of neurons, so identical axes, we see that the correlation pattern may look a little similar, but it's not quite the same. And the same thing is true for pentane-dione. And this level of similarity is expected because these are all chemosensory tasks. But I want to emphasize that, that these correlation patterns are more similar across animals for a given stimulus than they are for a given animal across stimuli. And from that, we see that even these relatively simple chemosensory inputs evoke rich network level responses. In response to the presentation of a single chemical stimulus, large portions of the worm's nervous system are engaged with distinct brain-wide functional correlation patterns for different stimuli. So the question has to be asked then, how do these functional correlations stack up against the C. elegans connectome? So C. elegans is well known for being one of the few animals for which the wiring diagram has been completely reconstructed. And this was first done via serial electron microscopy by John White and colleagues back in 1986. And there are, broadly speaking, two types of anatomical connections between chemical synapses, where neurotransmitters may be exchanged, and gap junctions, which are direct electrical connections between neurons. And both types of these connections were reported with some measure of structural weight. A best guess as to the degree of contact between two neurons based on synapse number in the case of chemical connections, and based on contact patch area for electrical connections. And so the most naive comparison we can make between functional activity in the connectome is to ask, okay, two neurons are highly correlated in their activity. Are they also strongly connected in the wiring diagram? And so by plotting functional correlations against synapse count for all recorded pairs, we can assess the extent to which the anatomical connectome explains the functional activity patterns we see. When we do these comparisons, we find that the R squared when comparing the functional correlations to structural rate of chemical connections is 0.005. And the R squared when repeating this comparison with electrical connections is 0.028. So in other words, neither the chemical connections nor the electrical connections in the connectome significantly explain any of the functional correlations we see. And I think this is unsurprising for a whole multitude of reasons. For one, we don't know the sign of most of the chemical synapses in the wiring diagram, whether they are activating or inhibiting. Nor is synapse count likely to be a good proxy for synapse strength. And also the stimuli presented in this experiment are a very limited set of stimuli, and it's not a representative sample of what this animal, what this circuit is doing all day. And finally, as I said previously, this sort of comparison accounting for only first degree connections is the most naive one we can possibly make. So unifying these two views of the nervous system, anatomical and functional, is going to take much more than just a simple linear regression. It's going to probably take a deeper understanding of the properties of the synapses themselves, as well as more sophisticated mathematical modeling. And so our group is actually working with Srini Turaga and grad student Lu Mi with this data set to try and do just that. So using this data to build a constrained neural network model of the C. elegans nervous system based on the topology of the C. elegans connectome. And in this first experimental project, we've really only scratched the surface of what NeuroPal can bring to functional imaging in C. elegans. So the strain is already available for use by other labs. And also Elena, a graduate student in our lab, is extending this experiment to a much broader range of olfactory stimuli, hoping to uncover a comprehensive picture of neural representation at the interneuron level. So we hope that moving forward, NeuroPal will not only allow for more rapid and efficient screening of neurons in C. elegans under various conditions, but also enable new scale-spanning approaches to questions in C. elegans neuroscience. With that, I'd like to thank our many collaborators for their contributions to this project. Thank you for your time, and I'd be happy to take any questions you may have.
All right, excellent. So thank you very much, Albert. So we've already got a couple of questions, sort of two related questions actually that have been posted up. Are the correlation maps different if you average over time using only one organism instead of averaging over many? And a related question, how much variability is there between animals in the neuronal activity patterns?
Those are both really great questions. I would say that the variability we see across animals for these correlation patterns is quite high. We see stereotypy in the neurons that we expect are involved in the chemosensory circuit, but there's a lot of correlated activity in individuals that's kind of not related to the sensory task. And so I'd say the variability is quite high, but it's lower for sensory neurons and for interneurons, which we either know or suspect are part of those circuits.
Excellent. So now we do have time for one or two more quick questions. Faculty, the floor is also open to you. We'll give it just another moment. Sometimes questions take a few minutes to pop up. So here's another question. Can this data be drowning in noise? That means could some of the structure be coming from other neurons that are unrelated to the behavior?
So again, this is about the correlations, I'm guessing, and by extension, the connectome. So I would say that one of the benefits of averaging over animals is that you kind of kill all of the activity, all of the correlated activity that's not related to the stimulus delivery. That being said, I'm sure there's still some correlations which appear in the data which are not directly stimulus related.
And Albert, I had a follow-up question that I'll ask is, this imaging technique seems very powerful. I always wonder when we're looking at activity over a broad swath of a network, if we're to take how sort of coarse our lens is for interpreting activity. So here you're looking at some kind of filtered effect of the dynamic. So if you have very precise timing codes or things like that, you might be missing out on that. Do you have a sense of how much resolution you have in sort of being sensitive to activity in the neural network?
Yeah, that's a great question. So first about timing, I think that as with most GCamp labeling, you're kind of limited in the interpretation you can do when it comes to fine timing things, especially if as is the case for a lot of CLI instruments, we're running with the 6S, which is, gives us better signal to noise, but at the cost of much worse temporal precision. And so because of that, we don't want to read too much into any of the timing information in this data set. And then there's also the question of kind of like, what is the limit of spatial distinguishability? And to some extent, that is a consequence of the optics you're using. But like in this first project, we were very conservative with the cutoff that we used. So to make sure that signals from neighboring neurons weren't contaminating each other and things like that, one of the things we did is we ran all this data through a demixing algorithm. And we also just cut off any neuron pairs which were closer than 5 microns apart. And so I think going forward, both of those things can be relaxed a little bit, but in this first pass, we were very conservative in trying to separate out signals of nearby neurons.
Okay, great. So I think we'll go on to our last talk then of the morning. Again, I encourage everybody to stick around afterwards. We're going to be transitioning to the poster session, but before we do that, Margie and I are going to give a quick overview of how the spatial chat function works and how you're going to interface with the posters. So please don't just disconnect immediately. There'll also be an extended question and answer period in one of the spatial chat rooms with the speakers. So our last talk for the morning is going to be Mariela Petkova, also from Harvard, and she's going to be talking about correlative light and electron microscopy in an intact larva zebrafish. So Mariela, go ahead and take it away.
Sure. Thank you, Simon, for the introduction. Thank you all for being here, and it's a pleasure to be here. It's also great that everybody's sort of thinking about neuroscience at this moment. I will be talking about 2 imaging techniques, light and electron microscopy in whole animal. But what really I'm talking about is what this question really is about structure and function in the nervous system. And when you think about nervous systems, they're super diverse. Some of them are just like nets of a few hundred neurons. Some of them have 100,000 neurons, like in the fly. A million, or larger organisms like the human or octopus, they have a billion or a few billion neurons. Now, across all of this diversity, there is one common theme. And this common theme is that all nervous systems have to produce behavior. And what I mean by that is the animal has to take actions which are appropriate to its circumstances. Like, for example, this fly, which is about to get swallowed, it needs to be able to detect the incoming threat and to fly away. So how do we actually try to understand behavior, and how does the nervous system produce it? A general approach is to first figure out what the problem is. What is the problem that the animal is facing? And in this case, there is an object which is coming in, and the fly has to detect it. And then we come up with a model for this narrow circuit or a network which will solve this problem. And this is a very famous model. It's been around for about 50 years. It's a Recurrent detector. It's a motion detector. But interestingly enough, we have only been able to test this model against the actual biology only recently. And, as physicists, we can come up with any sort of model, but really at the end of the day, they have to stand up to the biology, the circuit that we see in the animal. And when we think about circuits, really probably what pops up in most people's minds is Cajal drawing. Cajal is the founder of what we call today modern neuroscience. And what you see here is what we'll call today a neurocircuit for detection of stimuli from individual system of a bird. And what you see are the sensory neurons, which sense the environment, and there is information flow from the sensory neurons deep into the brain. The arrows outline the flow. What you should notice is that, not all neurons are the same. They have different morphologies, and this is reflected in the drawing. There is also connections between these neurons, and one thing that today we can do is we can see those connections with technical electron microscopy. And there is one thing that is sort of misleading about this drawing. It makes you think that everything, all the information is feed-forward, flows directly, but really, as Albert pointed out, the connections have a sign, and the sign can be excitatory or inhibitory. And what we can do today is we can use electron microscopy to visualize the whole structure of a nervous system. And what you see here is an electron microscope picture of a synapse. This is where all the synaptic vesicles are from the presynaptic side are located. This is a chemical synapse. So the vesicles will be released in this cleft here, and the postsynaptic neuron, its membrane potential will change depending accordingly. Now, another thing that we can do is we can actually figure out what is the sign of neurons, and we can do that using transgenic animals. What you see here are a bunch of cells which have been labeled genetically as excitatory or inhibitory. And what I have set out to do in my PhD was to combine these two types of information in the same animal. I want to modernize Cajal's drawings and do that for a whole animal at the limit of what we can do technologically, which is about 100,000 neurons. You could see that this is informative, and it's raising a lot of interesting questions in the case of C. elegans, as Albert pointed out just now. But what I'm trying to do, what I'm trying to do is to raise the same questions in a larger animal, a vertebrate, which has slightly different behaviors and somehow is closer to what our brain is doing. And to do that, I'm combining two types of microscopy. So I'm combining light microscopy and electron microscopy. On the left, you see light microscopy images of the cells, which have been tagged as inhibitory and excitatory. And on the right, you see the rendering in electron microscopy of a zebrafish brain and a zebrafish. This is the head of the zebrafish animal. You can see the eyes, and the reason why you can't see through is because in electron microscopy, we managed to get all of the anatomy, all of the structure, all of the neurons, the membranes, the muscles, everything, all of that anatomical information is there. And what you're seeing here is a very low resolution rendering of the whole image data set. This is only at cellular resolution because we're talking about a cellular question. But the whole image data set spans a few orders of magnitude. We're going all the way from cellular resolution or animal resolution to level of spatial scale to synapses, which are on the order of micron, and vesicles, which are on the order of a few tens of nanometers. And we can see all of that in the data set. So this data set actually comprises a few terabytes of data. But for now, what I'll talk to you about is how do we combine these data sets? And it turned out that we thought this was trivial because this is the same animal. We have imaged it into different techniques, but we should be able to put back everything together. Turns out that there are no actual machine learning algorithms or registrations software out there which can do that for us. But we humans are really good at it. So this is an example of intensity-based objects so this is an example of the light microscopy data where you can see the cells as intensity-based objects. They're little blobs on your screen. And in EM, these objects are boundary based. You can see them as outlines. So you probably already see that you can match up cells from one view to the other view. And it's kind of easy for you to do that, but turns out that there is no software which can actually solve that. That's still an open question in image registration. We figured that a great way to do this is to combine what our vision can do and then fine tune it using automatic techniques. So with a very talented undergraduate student, we manually matched the two data sets with a few landmarks. We find fiduciary points in the two data sets. As you can see, the green dot, which is popping up on your screen, lighting up, this is one of those landmarks. And when it lights up, it means we've overlaid it to images, and that should be hopefully clear. After we have pre-matched everything, we are running, we devised a technique called, we use a technique called iterative point cloud matching, where we locally compared neighborhoods in the light and EM microscopy. And we proliferated these 700 manual landmarks to more than 8,000, which are distributed throughout the whole brain. And this was really useful, because now to convince you just how well the matching worked out, you can see the overlay of electron microscopy and light microscopy. The boundaries of the cells are images from electron microscopy, and the colors inside each boundaries are the excitatory or inhibitory cells. And what you can really see is that we haven't just achieved single cell precision, but really it's super single cell, because we managed to fit in every cell inside its boundary. And this is really powerful because now when we extract, we can go into the high resolution images and we can start extracting the circuits. We not only know how they're connected, but also what the sign of the connections are. And to give you a flavor of that, we have found about 23,000 excitatory cells and about 10,000 inhibitory cells, which we can color among all the cells in the fish. As I said, what you guys have been seeing so far are low resolution images. These are the cell blobs at cellular resolution. But we have all of these images in high resolution with 4 nanometer X and Y resolution and 30 nanometer in Z. And you can see these very same cells have their lot of structures involved with them, and in this so-called neuropil region, this mess here corresponds to so much structure, which is the place where all of the synapses and contacts between different neurons occur. So to bring back the point, what I've tried to do is, as an experimentalist, is I've tried to modernize our notion of a circuit, of a neural circuit or House drawing, and this is what it looks like today. And of course, this has been a very large effort to generate this data set. These are all of the people who have been involved and helped. And it's a collaboration between the labs of Florian Engert and Jeff Lichtman at Harvard, but also we've been working with Google Research, because the images that you all see require a lot more computational power. And this is Zachary, the student that I mentioned, who helped pre-match the data sets. So with that, I'm done and I'm ready for questions. Thank you.
All right, great. So let's open up the floor to questions. Were you able to classify the neurons by shape in any way?
Oh, that is a wonderful question. We are doing that right now as we speak. Maybe we can update at the end of the summer, but we are building classifiers to tell us, which will tell us what the shape of a neuron is informing us about the function. And in this case, we can check whether it tells us something, whether it's excitatory or inhibitory. Thank you.
Other questions? And the floor is open to faculty too. I have a question, Mariella. Can you get, is there a possibility to get more precise than excitatory and identify the particular types of neurotransmitters involved so that you might have some more potential functional classification then?
So the real power here is that we are able to not just guess, but we can actually see that the excitatory or the inhibitory neurons actually we have a control, we have a measurement for them. We can confirm our guess based on the shape with or when we look at the neurotransmitter type. We haven't explored whether we can tell other neurotransmitter types based on the shape yet, but that is actually possible in the fly. I'm not sure how precise it is.
And then what are the criteria for labeling a cell as excitatory and inhibitory? That was a question.
This is the criteria is that the excitatory neurons are the major glutamatergic neurons in the fish, and the inhibitory neurons are the major the GABAergic neurons, which are the major inhibitory neurons in the fish. We are missing a few of the minor subtypes of excitatory and inhibitory neurons. This is the label.
So here's José Onushik. Can you tell a little bit more about the time resolution? Somehow got lost about it. How long take you to do an image and how can you resolve that?
So to take the imaging, so time resolution is a twofold question. First of all, those images are all anatomical stacks, so there is no temporal dynamics. To take the light microscopy images, it took us 3 hours of imaging. And to take the electron microscopy images took 2 months of imaging, 1 month of preparation, and now a few years of analyzing them.
All right, and we had a question of who drew the pictures on that slide that they're up.
I did that.
Those are excellent. I think many of us very much appreciate that. All right, so with that, I think we're going to end the speaking sessions. Thank you to Mariella and all of our speakers. There's a lot of virtual applause going on. You get to see me doing it, but everyone else can do it virtually. So folks, what we're going to do now is we're going to get ready to go over to the poster session. And we have a number of students who have very bravely agreed to give a virtual poster session. So we really want people to go over and attend and reward them with attendance and really, really good questions. So the way that we're going to do this is with spatial chat. And spatial chat is a functional chat room that is sort of like what we have just been using in Zoom. So I'm sharing my screen now, and Margie, I'm gonna go into room D just to show that room. So what you can do to navigate there, you have links for different spatial chat rooms sent to you previously in your registration. You can also find them on either the Georgia Tech or the Rice University Physics of Living Systems website. Here, if you go to the iPoLS meeting page, you can go to the poster presentations here. And if you click on this, you'll be prompted for a password. Margie's going to type the password into the chat. I don't want to say it here because we're also live streaming onto YouTube. We don't want to necessarily want it to be totally open. So any participant who's registered can participate there. On this page, there'll be a list of the posters. These are the posters that we have for today. So we have 19 posters today. They're separated into 4 rooms, okay? So you can find the list here. If you scroll down a little further, you can find the titles for the posters. They're listed here, okay? And then if you go down here, you have the links to the different rooms. So what's going to happen is when you click on one of these rooms, you're going to be taken to a program called Spatial Chat. This does not require you to install any new software. It's a totally in-browser page, okay? So you don't need to worry about having to install new software. Please give yourself your actual name so people know who you're talking to. Here I'm going to log in as Simon Swanberg. When you load up this page, it is going to ask you if you want to use your camera and your microphone. Please allow them to do so. And Chrome or something will ask for access to your camera. And then when you load this up, what's going to happen is, and I'm going to mute myself on this.
I think you need to turn off your microphone in Spatial Chat.
I just did.
Okay.
Okay, so now that you're in spatial chat, what you'll see is that you have a little circle, and we're still getting a little bit of feedback from somewhere. Okay, that should work now. So when you're in spatial chat here, what you're going to see is that you have a little circle and you have a little video bubble of yourself. And the nice function of spatial chat is that you can only hear people that are nearby your circle. So you'll see that over here we have Margie over here. And what I can do is if I drag myself over near Margie, I'll be able to talk to her. And you'll also be able to start hearing her. You can actually hear me echoing through her spatial chat a little bit. So this allows you to communicate only locally with other people. So you'll see that in this room, we have 5 posters that have been posted up, and you can drag yourself around the room like this. So if you just click on the background, you can click and drag the background around. I'm going to go up to my student's poster, Jeff's poster. So here, Jeff will be hanging out up here with his little bubble, and I'll be able to approach him. And if I want to learn about what's going on in this program, I can just, like a normal poster session, I can ask him, hey, Jeff, give me an overview of the poster. And then you can zoom in so that you can see better if you get closer, okay? So that way you can zoom in and out and get access. You need to be close. You'll be able to see when people are talking because they'll have a little ring appear around their bubble. So Margie, if you talk for a second, we can see Margie's ring light up here.
Hello, here I am in spatial chat.
Okay. And we're still getting a little bit of feedback because we're connected to the other systems. I'm not quite sure. I think it's because my audio is picking up my spatial chat audio coming through. So please do disconnect from Zoom once you're all set up here. So this is how we're going to do the spatial chat. So all the presenters are going to go to their posters. They're going to have their little icon hanging out here, and you just go drag up to them. And if you're a shy person, like I am sometimes at poster sessions, you can sneak up on people. You can slowly drag your circle closer to them and listen in to their conversation. And then when you get enough, when you've heard enough, you can speak up a little bit and start asking some questions, okay? If the presenters want to get an arrow, they can actually share an arrow image through the share image button up here on their screen. And then they can have an arrow that they can drag around their poster to highlight different things if they'd like. You can also toggle your audio and your video on and off here. So if you have a poor connection, you might want to toggle off your video. If you're trying to listen, you might want to toggle off your audio for a little bit, but make sure you turn it back on to talk to people, okay? So this is how spatial chat works. It's basically just like—
What's the password that we need for that?
It's for the Georgia Tech website. It's 2020 PoLS with an exclamation point. It's in the chat for the feature. And I would also add, we're going to ask that you guys social distance, even though it's virtual, because if everybody clusters on the posters together, you're going to hear the other poster presentations. So if the presenters can move to the edge, I posted a video on the YouTube channel and sent the presenters the link about how to make an arrow and where to be on your posters. So hopefully that will help y'all space appropriately.
So for example, Jeff is hanging out over here now because he's on the socially distant side of the poster, right? So he's not close to this poster.
It would be good to just mail this information to everybody because I'm...
So we're going to mail out everything in just a second. Let's just finish the discussions for just a moment.
It is in the email from yesterday. In the meeting update from yesterday, the password and the chat rooms are there. I will send it out again in 2 minutes.
What else? Margie and I will also hang out here on the Zoom channel. If anyone has difficulty getting access to the spatial chat or needs any help learning how to use it, please go ahead and let us know. And we'll be hanging out here and we'll be able to help you troubleshoot. And then we're going to go hang out at the posters as well. So the one other thing is that speakers can import videos like Jeff is doing here. So you can, if you use the share screen or share video option up here at the top, you can click in a video or you can share an image, and then you can drop that in and use that as part of your poster too. Most of the poster presenters should already know that. So now if there's, we're also going, there's also a spatial chat room for the speakers. So the speakers from today's session will go to a spatial chat room and you can go interact with them. Again, I encourage them to spread out a little bit. So we can already see that there's a fair number of people coming in here. So I'm going to shut up and let people thank everybody for a great morning session. Let people go to their, to the poster sessions. And then if there's any general questions here in Zoom, Margie and I will answer those.
And there is still the speaker room if y'all want to do that too.
And I'll post the links to the spatial chat rooms in the Zoom chat as well right now. Thank you all.
Say thank you, Simon. Say thank you, Simon. Say it. Ah, now she's shy. Okay. Thank you, Simon and Margie.
Thank you, Simon and Margie.
Thanks, Simon and Margie.
And all speakers.
And all the speakers, great talks.
Yes, indeed.
Yes, especially thank the speakers. It's so difficult to start to engage with these things in the new virtual format, but when it works really well. And I really enjoyed the talks.
I thought it worked. I thought it worked terrific in my limited way I could absorb it. Thank you, guys.
The spatial chat room links are in the chat now. You can click on them there and go directly to them. Margie, I didn't have the one for the speakers. If you can post the link for the speakers there as well.
Speakers up there too.
Jose or Peter.
I never found those passwords. Maybe I got the wrong emails. That's why I'm asking to resend.
Okay.
There's the password, Jose.
The password's right there in the chat.
Where? Which chat? I go to the chat. I don't see anything. Where? Oh, now I found it in the end.
Yeah.
There you go.
I wish we could have somebody virtually walk you over there, Jose, but
I can do it. It's just I couldn't find this password any other place. She claims on the thing yesterday. I haven't found it. That's the problem.
You can also just directly go to the poster rooms now using those links in the chat. You shouldn't even need to go to the website, but if you want to see which posters are where.
That's right. I want to see which posters are out there before I go there. Great, thank you.
Thank you.
Oh yeah, I can hear you.
The poster lists are also on the Rice PoLS website. It's everywhere.
It's everywhere.
So I'll hang out here. If anyone else, we still have a fair number of attendees here, attendees that are still here. You're either outing yourself as not really being here, or you're still joined elsewhere. But if anyone has questions about how to get in, please, I'm going to enable everybody to talk, or you can raise your hand and you can just ask questions if you need to. Okay. But please do join the poster sessions. JC says he's reading the poster sessions. That's fair, JC. I knew you were there. There's a link now to the poster session presentation that Ahmed post. Thanks for that. That's great.
Is there going to be another room for the speakers, you said?
Yeah, there is another room for the speakers. Margie's going to post that link. I'll see if I can find it. Hold on, too. It's the same link as yesterday, but I had the wrong page up.
How are you?
I posted it in the chat.
Great, thanks, Margie. Yeah, there's the PoLS speakers. Yep.
Thank you very much.
Really good talks, you guys.
Gabby.
How are you, Esteban?
Hello, Jose. How are you?
I'm fine. Trying to figure out how this thing works. Complicated.
Yeah, I know. So right now I'm using a mouse and it's making it easier. If not, then just zoom in and zoom out. But yeah, it's complicated. It is fun.
The email is off. You should have it soon.
Margie, are you there?
I am here.
I can't get into any poster room.
Does it give you any kind of message? It may take a while to load because the posters take a lot of bandwidth.
Yeah, Dave, it took mine a minute to load, but now I'm in and I'm chit-chatting with various people. Although, Margie, I'm noticing that some of the poster presenters are not in front of their posters. For example, Average from Bob Austin's group is not there, and a number of people are trying to find him. I don't know how we let everybody know.
I have been in repetitive contact with these people. I don't know what more I can do.
Okay, then that's as much as we can. There's no more bully pulpit we can use. I can write a note to Average. I know him and his advisor.
It may be that they're looking at somebody else's poster, or I am not seeing emails from people who are freaking out about not being able to connect. So I'm trying to get, I sent the email out, but I sent it through the wrong server. So I have to resend the email with all the rooms and all that. And then I will.
To confirm that the rooms are open, I've been able to access them. So you can get into them. You might need to also double check and make sure that you haven't had a browser pop up that's asking you for permission to use your microphone and video. If you haven't clicked yes on that, it may be stalling out loading up the room. So
Oh, it does ask me that question. Let me see.
So you'll need to respond to that question one way or the other before you can enter the room.
Simon, this is very cool. Thank you for finding this.
Oh, yeah, for sure.
Well, Margie's the one who really did all the hard work to set this up. It's a bit of a pain to set up and get everything going.
Of course.
But the posters look really nice.
That goes without saying.
Thank you very much, Marguerite, for this. Thank you very much.
And if you guys get on the spatial chat rooms, you may have to eventually leave Zoom because we're going to get a fair bit of feedback through here if you're connected both through Zoom and the rooms.
I'm gonna get off. I'm gonna get off Zoom. It's been an honor and a pleasure. Thank you all.
Go hang out at posters.
Before you get off, Dan, are you already off?
No, I'm here.
So are we going to do it the same way tomorrow also? The only thing I was thinking is, so did we only have the speakers and Margie and the moderator?
Simon and Margie have been running it. I've had some family stuff, so I'm kind of in and out here. So direct all questions to.
Okay, to Margie. Yeah, I was just saying that I kind of liked if a bunch of faculty were on the panelists, but they still don't, they can still be not allowed to ask questions until at least students have asked some questions. But I was just wondering about that, but we can talk about that later.
Peter, I think it doesn't make too much of a difference because you can enable any of the attendees to be able to mute and unmute themselves just by clicking on them and saying allow them to talk, and then they can toggle on or off. So the reason why you can unmute yourself and talk right now is that earlier on, Margie or I clicked that for you. But you mean you'd like other faculty members to be able to see the sort of executive panel?
I mean,
we have today.
Yeah, for me, it's like, at least it feels like you're attending a meeting when you can see somebody is there. Otherwise, it feels like, you're just listening to something passively. But I guess practically speaking, it doesn't make a big difference. So, yeah.